Yeah, how about that? All right. Yeah, so this was work that was led by Dong Yuan, and unfortunately he couldn't be here due to visa reasons. But I'm happy to walk through it, and I'm hopefully familiar enough with the content to answer any questions. So just a bit of background overview for the talk. We'll go through some mild background and motivation for the problem, and then describe what the model is and how we perform inference, and then go over some results. So I think at this point in time, it's kind of, there's no need to hammer home the point that there's just a massive amount of data that's out there, and with respect to covering different modalities of like biological processes, right? So we have genetic data, we have epigenetic data, we have transcriptomics, we have proteomics, there's metabolomics, and something that's kind of core in terms of inferential procedures that's been throughout all of bioinformatics, amongst many fields, is really this notion of like matrix factorization or factor analysis in some way, shape, or form to get at broadly like latent structure, right? So some shared structure that is maybe kind of explanatory for many different aspects apart from some like additional kind of like noisy observational process. And so like one exciting area for leveraging either matrix factorization or latent factor analysis is with this more recent MPRA or perturb-seq style data. So just like very briefly, what this data entails is like you have these large-scale experimental screens. Well, they'll knock out maybe hundreds of genes, or now from some of the larger data sets, thousands of genes in a single-cell manner. And what one can do is get an estimate of the effect size of knocking out a gene or knocking down a gene on downstream expression, right? And so what that gives us is some sort of like loose sense of kind of the regulatory consequences if we were to just sort of disrupt this gene in some way, shape, or form. And so one thing that we'd like to be able to do with this kind of data is, again, to make maybe an assumption that the underlying structure that can sort of generate this observational process is that there's some shared latent representation with like kind of noisy, either noisy biological stuff on top or noisy technical things on top. And so we'll come back to that later. But if we wanted to just throw any kind of dimensionality reduction or latent factor analysis at that, there's some limitations, right? And that the traditional approaches like PCA and SVD can be very scalable depending on how the particular implementation works. There's iterative procedures to do this where you don't need to sort of pre-compute a genetic covariance matrix. You don't need to compute all the possible latent factors. You can just specify some K, and it can spit it out for you. The other aspect so that's a benefit, but one limitation of these procedures is that while straightforward, they can provide limited interpretability, right? So they, by definition, tend to output dense loadings, and so sort of understanding what they're doing can be challenging. Kind of on the flip side of that, you have things that are like sparse PCA or PMD, where you have these latent factor models that are able to compute sparse loadings or sparse representations, right, that improve interpretability, right? We can sort of shut off the things that aren't being contributed to or explained, and so we can kind of take away what's happening, but there's a computational cost, right? These things tend to be expensive to run. And so something that can like sit in kind of the middle and between those two a bit, you have things like probabilistic PCA or like empirical Bayes matrix factorization that can be very scalable, but depending on like the particular model might not sort of promote sparsity, right? Again, so like none of these methods above sort of immediately provide a framework to select the features while being scalable. Right, and so what we'd like to do is how do we address the limitations of things like interpretability while keeping the benefits of computational speed. Oop, okay. And so just before we get into like what we propose, just a quick review is there's some recent work and very fast sparse regression models called SuSiE, where there's this notion where if you have a sparse linear regression framework, here represented by y is equal to x BL plus e, where you can decompose the effect sizes as L separate effects, right? And so the way the fitting procedure for this type of model works is that it's very, very fast, right? It's essentially linear or quasi-linear. in terms of the input of the data. And so it's also probabilistic, and that not only is it sparse and it's quick, but we can sort of quantify the uncertainty on what it's selecting, right? And so this is kind of in contrast to like purely optimization-based sparse objectives, and that here we actually have a means to sort of kind of do hypothesis testing, right? Whereas in something like if you just have like an L1 penalty, you might need to do cross-validation or something to sort of quantify how sure we are about what's being selected. And so those are something that we call a posterior inclusion probability. I'll explain a bit more about that in a second. But what the key thing to take away from what this model is doing is that, so BL, which is a vector of effects, can be decomposed as the sum of 3 independent effects, and here if L is 3, right? And so if we had 3 total effects, right, that's saying like 3 features out of all of our features were being selected, we could rewrite that as a sum of these single vectors, right? And so this is the notion that underlies SuSiE. And this is kind of the key idea that we extended to PCA, is that we'd like to be able to leverage this notion of the sum of these smaller effects into this matrix setting to have a fast, sparse solution to PCA, essentially, or factor analysis. Right? And so our goal is to select only L features for each factor. Here we have K factors. And this p-dimensional data. And so if we have a matrix X, which is n by p, and we have a Z matrix, which is n by k, right? So for each observation, there's kind of this lower level k-dimensional representation. And our loadings, which is a k by p matrix, is going to be our sparse matrix. Let's see. Oh, that's not really showing up so well. So here's kind of like the core notion of the idea, in that we can decompose our matrix as just this kind of typical matrix factorization or matrix decomposition, just like we've seen in some of the talks already today. But what we've done is say that this matrix W, so maybe it's a bit easier to look at pictorially, but if we have this matrix here that represents our loadings, we can decompose each row as kind of like a SuSiE scenario, right? So each row then can be represented as the sum of L effects. All right. And so, so that just really quickly, maybe more formally, right, is that each row can be represented as that, and then each individual row will have its a prior over the actual effect sizes. When there is an effect, it has a normal prior, and then what is being selected can be kind of like a uniform prior over the features. But certainly that can be generalized through some additional annotations of the data. All right, so that's the model, but how do we perform inference? As you might guess, if we wanted to sort of fit the posterior for this model exactly, that's just intractable. There's no way we can do it. And so we leverage variational inference. And so essentially, we can think of the fitting procedure as kind of generalizing how SuSiE works, but over these latent features. And so for each observation, we have this latent feature vector, and we're able to keep things to scale to be like kind of quasi-linear with respect to the data. And so that's how we fit our latent factors and our loadings. But additionally, we also have these hyperparameters that govern sort of the prior variance on the effect sizes. And so to learn those, we just do kind of like empirical Bayes. Similar to what was alluded to in the Vipers talk, where you can learn some of the prior parameters like over the data. Here, that's kind of what we're doing, right? You can imagine fitting the posterior for each of these. You do that for everything, and then once that's been done, you go back and do an additional kind of like maximum likelihood step. And so once the model converges, we can also compute this notion of this posterior inclusion probability for each latent representation. And what this provides with us is a way to quantify the uncertainty or the probability that some feature is captured by that particular latent factor, right? And so I think this is kind of like the key benefit of using a tool like this. Is so now not only do we have the sparse representation, but we can also then say what's the probability that this feature is in this particular latent factor. So the first thing we sought to do was to, certainly we can write out all this math, but we sought to quantify how well it does in simulations and compare it with some additional existing works. And so we performed simulations where we generated data that have sort of a structure that looks, kind of, how do I do this? we can see it decomposing into more or less like 4 factors, with effects being sparse within those factors. And then we compared our tool versus sparse PCA and this other tool called empirical Bayes matrix factorization, which is another sort of sparse probabilistic factor analysis method. And then we had 2 metrics that we looked at. So for folks familiar with kind of like factor analysis or like matrix decomposition, you can only learn things up to scale or rotation. And so it can be, you can have like an arbitrary rotation in your latent factor space and still have the exact same likelihood. And so we didn't want to penalize ourselves or other tools by trying to see, do we capture exactly the latent factor when it could be that it did, but it just rotated it slightly, right? And so we have this metric that accounts for that. So it's this Procruste's norm. And so all it is that, if you have this sort of true existing set of loadings or factors, for each tool that has this predicted loadings or predicted factors, we just see essentially up to rotation and scale how well does it capture it, okay? And then similarly, we can also just look at like kind of predictive performance, right? If every model is able to compute loadings and compute factors, we just see, does it predict the data well? And so the first thing that we looked at was just, if we were just to run our tool, we wanted to compare which performs better in terms of selecting features, right? So if plenty of other tools that do just pure optimization with like in a non-statistical manner, just kind of minimizing some loss. If we wanted to say like what is being captured by the data, what features are in each factor, right? We could just simply take the loadings, right? We can take the weights. And so what we did is we just said, fine, let's take the weights that are the absolute value of the weights and see how well at various cutoffs, like is our sort of classification error with respect to the true positives or true negatives. And then we're comparing those to the pips, right? Our probabilistic kind of notion of whether a feature is being represented in the data. And one thing that we see both in terms of capturing either the true positive signal or sort of rejecting the true negatives is that the pips overwhelmingly outperform trying to use weights themselves to select features, right? And so I think this is just, again, kind of motivation that it makes sense to look at things through a probabilistic manner rather than just some arbitrary weight to say that something's non-zero or has a big weight. It's probably being selected. Okay, and so now getting into the actual direct comparisons with other approaches. So SuSiE PCA outperformed all of the other approaches across a couple of different parameter settings in terms of recovering the loading matrix here. And so here we're looking at this kind of Procruste's norm. And so SuSiE PCA is in yellow, lower is better, and the other tools, sparse PCA and empirical Bayes matrix factorization, are in blue and red respectively. And so you can just see by varying the sample size, by varying the number of features for a fixed sample size, by varying the latent dimension, and the number of signals that are contributing to each particular factor across the board, that SuSiE PCA beats the other approaches. Similarly, if we kind of look at the same setting, but instead of looking at this Procruste's norm, just like how well does the model predict the data, again, across the board, we see that SuSiE outperforms the other two approaches. So again, across sample sizes, across the number of features that are in the data, across the latent dimensions, and across like the actual number of features that are within each factor, SuSie's beating the other approaches. So one thing that's typically challenging when it comes to factor analysis is how does one choose K? And so what we wanted to illustrate is how robust is SuSiE PCA itself to the specification of K and this additional parameter that users are required to specify L, right? And the L captures how many effects are represented in each latent factor. And so here what we have on the left is that you can increase the L that you specify to SuSiE. And I want to say, so the true is, what am I looking at? I want to say maybe the true is 40. Oh, right, yeah, L is 40. So the true here, so if L is underspecified, obviously the error is going to increase. But importantly, if you overspecify L, the error doesn't increase, right? And the reason for that is that SuSiE PCA, since it's learning these prior effect sizes, variances, they're just kind of shutting off the effects that aren't explaining anything, right? So you can overspecify L, and the model will learn to just shut off those things. And then similarly, on the right-hand side here is, let's say that L is correctly specified, but we misspecified K. And so the true K here is 4, and then we see that if you overspecify, that the overall performance doesn't drastically get worse. And again, this is because the factors are essentially shut off. The L effects within that component won't be explaining much in the data, and so the model just turns them off. Whereas we see that the other approaches can be quite sensitive to this in terms of their error. Let's see, how am I doing on time? Oh, we still got like 10 minutes. Okay, great. So now that through simulations we've demonstrated that using PIPs is a better means to select features, that SuSiE PCA outperforms these other approaches across a couple metrics and different parameter settings. But in addition to that, SuSiE PCA runs up to 50 times faster than these other two approaches. And again, because it's more or less like quasi-linear, or with respect to the data, maybe borrowing some additional constants from the number of latent factors. And here, what we're showing is just that if we increase L, like the true number of signals in the model, the runtime really just scales somewhat linearly with respect to that. But in all cases, we see that there's a large gap in terms of the runtime. This measurement's in seconds, and so for this particular simulation, everything's kind of fast. If we want to take a step back and say that qualitatively in this simulation or setting, everything's fast, but we'll see in a minute like how that actually applies to large-scale data. What is this? Oh, sorry. I think there's a, and this is again just the same setting, just as we increase the feature dimension. And then again, across the number of latent dimensions. Sorry. OK, we can just kind of skip ahead. All right. And so now that we've gone through simulations to sort of convince ourselves that our tool is doing the right thing, it's faster, we wanted to apply it to real data. And so what we did is we downloaded some perturb-seq gene expression data from Upogo et al. So the actual data consists of about 310,000 it's not perturbations. Essentially, it kind of boils down to they had like 2,000 genes that were perturbed, but there were multiple guides. So there's like repeated perturbations depending on the gene. And so we did some pre-processing where we collapsed the cells. Oh, sorry, there's the number of cells. So we had 310,000 cells across this many genes. And what we did was collapse across the cells into a pseudobulk to get just 2,000 versus observations versus these 8,500. And this, for now, is really just to deal with memory constraints. So the tool isn't really set up to run on sparse observational data. That's something that we plan to address in the near future. That won't take much effort, but just in terms of what we've done so far is it can't handle that for memory reasons. We did some pre-processing where we regressed out kind of just mitochondrial percent and then some germline group for the normalized data. And we input these into SuSiE PCA with k equals 10 and L equals 300. And so just to look at runtime, we also ran the other tools on this data. And so SuSiE PCA ran in about a minute. EBMF took a couple minutes to run, and we see that sparse PCA took closer to 20 minutes. So already, while we might not have sort of a 50 times speed up, we have on the order of anywhere between 2 to 20 times speed up. So just looking at kind of like the heat map of the loadings to see like the effect sizes that are learned across this. So on the left-hand side, what we have are the loadings across the 10 factors. And again, so the scale is a bit different from the one on the right. And the reason is that if we match this to this scale, this entire sort of matrix would just end up looking white, and that the effect size would be too small. And so let's just keep that in mind. But what we do have here is kind of like a much sparse, I should say, a very sparse representation of the data, where this, while the scale is a bit different, but here it seems to pick many, many more genes that are being reflected in this lower dimensional space. And so one thing that we could do to say, well, fine, we ran this tool on this data, what is exactly that we're learning? And so we wanted to say, for a given latent factor, are we essentially getting kind of gene networks, right? If we're getting sort of gene regulatory networks that if you knock out some gene or group of genes that they're all have this shared correlation structure that's sort of independent from other regulatory networks that just can be captured by the general perturb-seq process. And so the first factor that we pulled out is essentially just ribosome. factors. I think this might be unsurprising given that probably a lot of the DNA is still going to be reflected along those lines. And so if we were to take the loadings here from this factor and then plug them into something like GSEA to get what pathways are enriched, obviously we get a bunch of things that are related to ribosomal pathway. There's some evidence of coronavirus disease and a couple fewer pathways that have some mild enrichment. But where things can get interesting is here in factor 4, we see things that are highly enriched for DNA replication. We see some perturbations that reflect some genes PHB1, PHB2 that are both just happen to correspond with aging, proliferative metabolic disease. And so we see these enrichments here. We have a huge enrichment for DNA replication, mismatch, repair, steroid biosynthesis, and a couple of others. Importantly, something to note, just as kind of like a control, in both scenarios, we have these non-targeting genes. And so these essentially kind of I think just sort of spike-ins in the model to get a sense of where expression is at. And the loadings for those tend to correspond to more or less null. If we want to compare our findings to those that are spit out from sparse PCA and empirical Bayes matrix factorization, it founded similar components with respect to latent factors, but the loadings, if used to go and identify pathway enrichment, it has much, much less power compared to those from SuSiE PCA. And so while it does find ribosomal pathways and similar pathways like coronavirus disease, in either tool, the log fold enrichment, I think, is on the order of about, let's say, maybe 50% as much as the strength of our tool. And so we wanted to sort of assure ourselves that our findings are somewhat robust to how we selected K, how we selected L. And so we reran by respecifying K, respecifying L. And so one simple analysis, if we were to increase K, we can then say how often is it that the learned factors are correlated with one another. And if they're correlated, it's kind of recapitulating sort of the same biology. And then that's some evidence of it just being redundant, that the model isn't necessarily needed in the factor space, right? And so if we were to increase K beyond 10, we started to observe more correlated factors. And so we just, as a heuristic, felt that was kind of unrepresentative or not necessarily needed. And if we were to sort of change L, that it would have some effect on the number of genes that are associated, but it was largely only those in the top 2 latent factors, and that the lower, downstream factors didn't seem to be as sensitive to that. And so here we do see more or less the top factors still capturing the same broad pathways when plugged back into GSEA. Okay, so our tool's already out. Folks can download it and run it on their own data. So it's available at GitHub under our lab page called just SuSiE PCA. It's a Python tool. It leverages this library, this really fantastic library that Google developed called JAX, where JAX can do sort of just-in-time compilation of the Python code down to C++. So you get to write Python, high-level Python code, and for free get C++ code that can run on GPUs or CPUs or now TPUs, if you have an account to run things on TPUs. And additionally, JAX can do things like auto-differentiation and gradient analysis. We're not leveraging that for this project, but I don't know, that's just my short sales pitch for JAX too. So just in summary, SuSiE PCA outperforms sparse PCA and empirical Bayes matrix factorization in terms of selecting true signals and simulated data, estimating the loading matrix, and prediction accuracy. We found, again, that it was mostly robust to how L and K is specified. By leveraging variational inference, we're able to get 20 to 30 times faster inference compared to existing approaches. Applying it to real data, we found that SuSiE PCA identified distinct biological functions with different components and improved power when using the loadings for downstream gene set enrichment analysis. And as I mentioned, it's available on our GitHub for folks to use. And so, obviously, I want to thank Dong, who can't be here today, who led not only sort of the mathematical derivations for this work, but also did a great job implementing it and running all these analyses, as well as all the other members in my group who kind of through discussions contributed, as well as Stephen Gazal's group, whose lab is right next door to ours. So thank you so much, and if you have any questions, happy to do my best to answer them.
Time for one question.
I have a comment.
Nice talk.
Thanks.
I was wondering, actually have many questions, but I'll just ask you one.
Sure.
Okay? So in one of the data sets that you used, you took cancer DepMap or dependency map data, and there was a bulk RNA. So you had single cell data that you converted to the bulk data for the matrix.
Oh yeah, so it's they had single cell perturb-seq data that we then kind of, for computational reasons, collapsed down to bulk.
So my comment or question there is that, so of course you have single cell data which is at a higher resolution and for this. So how would it work if you subsampled, retained single cell, subsample the data? Would you get similar networks? Or,
Yeah, that's a great question. That's something that we could definitely explore. I think that'd be a good way to actually assess, the resilience of the tool to just sort of natural sampling. Yeah, that's a really good idea and something we should do. I think, long term, it'd be great just to run it on the actual single cell data. There's some nice, another small plug. Jax has this nice means to just wrap a function in another function and make it just translate to sparse matrix representation. So I think that's something we want to explore. But I think just the simple, your nice suggestion about downsampling that would be a really good one.
And it would also probably display the robustness of the thing, resilience and robustness as well.
Mm-hmm. Yeah, really, really great idea. And then happy to chat more offline. Thank you. Cool.
Thanks again.
Yep, thanks.